interesting time. So it's my pleasure to have another fluid seminar given by Dr. Maciej Lisicki. So even for someone Eastern European like me, I have trouble with Polish names.
In fact, this was perfect pronunciation.
I have followed some of his work. So he did his PhD in Poland with Bogdan Cichocki. And it turns out that actually we are somewhat academically related because my advisor at Yale was also student in the same group and worked with Bogdan Cichocki. So that's how we end up having a similar passion for low Reynolds, Stokes flow, and multiples and spherical harmonics. And after his PhD, he did a postdoc, and he spent a couple of years with Eric Lauga in the Mecca for Physics in Cambridge in the Department of Applied Math and Theoretical Physics. And a couple of years ago, he started his permanent professor job at Warsaw. He's visiting on sabbatical in University of Pennsylvania on a Fulbright scholarship and traveling around and sharing his exciting research on swimming. So welcome.
Thank you. Thank you very much for the introduction. Right. So I'm going to tell you one or two stories about some aspects of microscale swimming that are related to the coupling between fluid dynamics and elasticity, and hence the title Elastohydrodynamics of Microscale Swimming. And this is a tale of tales because microscale swimming is mostly about having a tail and wiggling a tail in a fluid. And so I'll start with a broad introduction about this wiggling motion of tails. That worked well. And so the question we start with is how do microorganisms move? And so if we look down there, there's plenty of different shapes and different motions that these microscale organisms do. And if we look at a couple of selected examples, so most of these microorganisms, they look like tiny, hairy potatoes or potatoes that have only a few hairs. And so the main difference that there is a variety. So for each of these species, you can describe in detail how they move. But the main line of division is about here, that these despite different morphologies and physiologies, there is the major division biology-wise is into two groups of prokaryotes and eukaryotes. And prokaryotes are mostly bacteria and archaea, and this is these five organisms here, and eukaryotes are, examples of eukaryotes are these two here. And the main difference is in their biological structure, which determines how they move. And so bacteria and archaea, so prokaryotic organisms, are much simpler. And so they all have these helical or slender filaments. They move in the fluid, but these filaments are fundamentally different in the case of prokaryotes and in the case of eukaryotes. So this is a prokaryotic flagellum, and the eukaryotic flagellum is drawn to scale here. So the prokaryotic flagellum is a very thin structure, which is basically a polymerized peptides. And so it's a slender structure that is completely inert. So an inert filament that is being attached to the cell by a rotating molecular motor. And so there is an elastic part called the hook that couples the filament to this rotating motor. But in essence, the structure is very simple. There is the motor, and there is a helical flagellum that this motor can rotate. Now, in the case of eukaryotes, and examples of eukaryotes are algae, ciliates, sperm cells, and we are examples of eukaryotic organisms. Eukaryotic cells are generally more complex, and they have a cell nucleus. And so in terms of flagella, an eukaryotic flagellum is much thicker, and it has an internal structure called the 9+2 structures, in which you have 9 pairs of microtubules on the circumference of this cross-section of the filament and a central pair. And so these are like a scaffolding that gives the flagellum rigidity. And in between these bits of the scaffolding, there are motor proteins called dyneins, and these proteins can walk along the scaffolding and thereby induce active tension along the filament. And so, whereas these are inert, these filaments are inherently active, as in there is active tension distributed along them. And so if we look at prokaryotes first, so how they move, the working horse of the field is the E. coli, bacterium everyone looks at. And if you look at this merry gathering of E. coli, well, they mostly swim along straight lines, but then sometimes something happens and they turn. And this something, because of that, their motion has been termed run and tumble motion. And the underlying mechanism for that is the following. So if you have an E. coli swimming along a straight line, like this colleague here, this one. Well, when it's moving along a straight line, we call it a run, and all of the molecular motors are rotating in the same direction then. And in result, there is a hydrodynamic mechanism that brings all the flagella together, and they rotate in the form of a bundle. And so this results in a straight motion. And then when suddenly one of these motors starts rotating in the other direction, well, that leads to a stochastic unbundling of this bundle. And this stochastic unbundling leads to a reorientation of the bacterium. And this we call the tumble motion. And after that, all the motors start rotating in the same direction again, and the flagella bundle together. So this unbundling is responsible for the random reorientation of bacteria. And so this is quite a complex motion already, whereas the interactions between these flagella are mediated by the fluid. Now, the case of eukaryotes, well, eukaryotes are much more richer in shapes. And so this is an example of a group of eukaryotic organisms that are ciliates, and you can see the ciliate diversity chart here. And probably, well, most of them have what they have in common is they have these hairy bits. You can have a hairy surface here, or you can look like a hairy potato altogether. And probably the example that is well known to almost everyone, or at least kids in the Polish school, are always faced with this paramecium here. Paramecium is the standard example of a eukaryotic organism. But paramecium is all covered with tiny cilia. And these cilia, they can perform active motion because these are eukaryotic cells. And what they do, most of the, they can perform lots of different kinds of motions, but the standard one that we focus on typically is called a two-stroke motion, where a cilium, an individual hair on this potato, performs a power stroke, which is a motion mostly perpendicular to the plane of the cell. This is this reddish motion here, where it pushes the fluid along the cell, and when it's almost parallel to the cell surface, it performs a recovery motion. And so this is the power stroke and the recovery stroke. And it's sort of like a surface-constrained breaststroke motion of an individual cilium. But then they have plenty of these cilia, and what they do is they coordinate the motion of these cilia to form these so-called metachronal waves. And so by achieving the synchrony, they systematically pump the fluid along the surface of the cell, so in a manner resembling a Mexican wave at a stadium. And so they push the fluid along, and in result, they can stir the fluid around or achieve propulsion. And so this is another example of a ciliate, Tetrahymena, and this is an algal colony of Volvox carteri. Well, again, it's not a single cell, but a multicellular organism composed of cells each of which has a pair of cilia. And so this is another hairy ball that achieves stirring of the fluid by coordinated motion of the flagella. And the coordination of this motion is partly in result of biochemical processes inside the cells and partly because of hydrodynamics. The ciliary stirring and pumping is actually more omnipresent than we think. So this is an example of a human fallopian tube, another eukaryotic organism in which then synchrony is achieved on a multicellular level. In the fallopian tube, in the reproductory tract, we have tufts of cilia that transport the mucus, and so they are surrounded by cells that secrete this mucus. And so the transport of mucus in the cilia in the reproductory tract is achieved by coordinated motion of these tufts of cilia. Same in the cerebrospinal fluid and same in the lungs. So different types of mucus or different types of a fluid, but the same mechanism. And so as a physicist, I tried to look for universality rather than diversity in this whole context. And so the underlying or the common denominator of that is that everywhere there's an aqueous environment. And so all these systems have to play by the same rules of small-scale hydrodynamics. And because all these flows typically happen in microscale, so on the length scales of micrometers and with typical velocities of micrometers per second, then the Reynolds number that gives you the character of the flow, if you use these values of the velocity and the length and the viscosity of water, then it turns out that the Reynolds number, which is the relative importance of effects of inertia to viscosity in the fluid, is much smaller than 1. Which means we can completely neglect the effects of inertia in favor of viscosity in the fluid. And so the Navier-Stokes equation that we would typically have to describe the flow of fluid in the system simplify greatly to these Stokes equations, which are complex enough. But the advantage here is that in these flows, there is no inertia, there is no turbulence, and the equations are nicely linear, as you see here. So they are linear in the velocity. And so there are a couple of consequences of this fact. And one of the consequences of this fact is that on these time scales and length scales, there is no time derivative in the equations, which means that these equations are instantaneous. So any disturbance in a fluid that happens because of motion of a cell is instantly communicated to the whole fluid. So everyone feels it at once. Now, another consequence of this linearity of Stokes equations is the fact that, in fact, here we have the dynamics that Aristotle thought would govern the world. So if you're acting on a body with a constant force, it will move with a constant velocity. So, well, this is not quite the world of Newton's second law, where you have a constant force, you should have constant acceleration. In this case, you have constant velocity. And another consequence of this fact is that the drag force that the cell moving through the fluid feels is proportional to the velocity of its motion. And the third property of Stokes' flow is kinematic reversibility. And to demonstrate this property, I have this little experiment, which you might have seen, but if not, it's really fantastic. So what we have here is an annulus between two cylindrical surfaces, and it's filled with glycerin just to have a fluid with a relatively low Reynolds number. And so what we do now is we insert some colored dye into the fluid, and we are going to turn the inner cylinder in the clockwise direction. And so we're going to mix this fluid. And this mixing motion, well, we're going to give it a stir, a couple of turns in the clockwise direction, and we smear this fluid inside this cavity. But now, after 4 turns, we're going to give it 4 turns back. And so this is analogous to stirring your tea, where your tea gets sweeter all throughout, except it's not. Because we came back to what we started off with. And there is a bit of smear due to molecular diffusion, but we came back to square one. And this is illustrated, this has consequences for the propulsion of microorganisms called the scallop theorem. And the scallop theorem says that no reversible swimming gait can lead to self-propulsion. And so this hypothetical microscallop that would only open and close its shell would go nowhere, because this is a reversible sequence of motion. So whatever it would gain by opening its shell, it would lose by closing it back again. So this one parameter transformation wouldn't work. For the same reason, fish do not swim in a highly viscous fluid, well, in a viscosity-dominated world. So this explains why there are no microscale fish. Why do cells in microscale not look like fish? Because the strategy that fish use for propulsion is completely ineffective at small scales. Because whatever a fish would gain by flapping its tail to the left, it would lose by flapping its tail to the right. And so fish do not swim in honey, and microscale world feels like swimming in honey. So if you want to understand something about swimming in microscale, you should imagine yourself in a swimming pool full of honey. And life is a bit different then. Well, to illustrate that, you can do a macroscale experiment, and this is actually based on a wonderful YouTube movie or a video of G.I. Taylor from 1966 on the properties of low Reynolds number flows. You have here we have a mechanical model of a fish, which the propulsion is achieved by a rubber band that's wound inside, and that when released, it just flaps the tail of the fish, and so you can see the fish moving now in water. If you take the same mechanical fish and put it in a tank of golden syrup, which is a highly viscous fluid, then you see that this fish is not able to go anywhere. And so it will just sadly, deprived of nutrients, it will sadly die here. Now, if we take a mechanical bacterium, which is inspired by living cells, so we have these helical tails, and the propulsion mechanism is essentially the same, well, then you see that perhaps this would work better. And indeed, so by just rotating its helical tails, it achieves propulsion. And this is inherently related to the coupling between translational and rotational motion of these helical tails. All right, so there is more universality in these Stokes flows. And one important thing is that the flow field, if you have a particle settling in a fluid under gravity, then it's a motion driven by a constant force, the gravity force of the particle. And so it's like applying a constant force to the fluid, well, in this case, in the upwards direction. Then the flow field that you induce is presented here. And so the solution of the Stokes equations under the action of a constant force, we call it a Stokelet. And so this is a universal flow field, or far field flow, you would get if you drop a steel ball into a tank of glycerin. And it's moving then at a constant velocity because if it's moving under the action of an external force, external constant force, or the gravity force, then there will be an equal and opposite drag force to balance this external force. And so this balance determines the sinking velocity. Now, in the case of microswimmers, the game is slightly different. And one question we might ask is, how does the flow field look like in this case? And it turns out, well, if we turn to the analysis of forces acting on the swimmer, so this is an example of a swimmer. It's a model of Chlamydomonas, an alga. And so by flapping these two flagella or cilia in a sort of a breaststroke-like manner, it exerts propulsion force on the fluid. Well, so in result, there is a fluid reaction force acting on the cell in the forward direction. But then the cell starts moving at a constant velocity, u, and that induces a drag force, from the fluid on the, So there's a fluid reaction force, and another force acting on the cell is the drag force. So all in all, we see that there's a pair of forces acting on this microswimmer, that is the fluid drag and the fluid reaction force to its propulsion force exerted on the fluid. And so a swimming cell can be simplified, or at least if looked at from far away, as a pair of forces acting on the fluid. And so this propulsion balanced by drag results in this pair of forces acting on the fluid, and the corresponding flow field looks somewhat like this. And we call it the dipolar flow because there is a pair of forces acting on the fluid. And in fact, this is quite universal. So if we look at flows around swimming cells, well, for one beautiful experiment by Knut Drescher from 2011 is they measured the flow field around a swimming E. coli, so a bacterium, a prokaryote. Well, then you see that the flow field has this symmetry that there is one axis along the swimmer direction where the fluid is being ejected to the front and to the back and being sucked in from the sides. And if you look at a completely different swimming organism, an alga, so a eukaryotic organism with a completely different biological mechanism for swimming, well, it does this breaststroke-like motion, but if you time average this motion and you look at it from far away, you get this sort of a picture where the fluid is being dragged in from the front and the back and ejected to the sides. And so there is a universal character of flow in these organisms, and in fact, a generic flow field of swimming microorganisms. that we call a dipolar flow. And depending on whether you're sucking the fluid in from the sides and ejecting to the front and the back, then you're called a pusher, like E. coli. And if you're doing the opposite, so you're dragging the fluid in from the front and the back and pushing it away to the sides, you're called a puller, like this alga Chlamydomonas reinhardti. And so it's a universal feature of microscale flows, and all the organisms that move do have this. In the far field, they look the same. And if you take the full flow field and you subtract this dipolar term, well, you get almost zero, which means that far away from the swimmer, you're very well off by just approximating the flow field by a dipolar flow. And close by, well, there are, of course, differences which are more quality dependent. All right, and so because most of that motion involves the motion of slender filaments in flow, now another important bit of information is to relate the fact that, well, what is the flow field induced in a fluid because of the motion of slender filaments in flow? And these filaments are of course, anisotropic. And it turns out that it's much easier to move such a slender filament through a fluid along its axis than perpendicular to its axis. And so if you have a filament that's moving under the action of a force, it's going to move, well, its velocity is going to be proportional to the force because it's a linear world where Stokes equations are linear, but this coefficient of proportionality is tensorial, which reflects the fact that the drag forces acting on such a slender object along its axis and perpendicular to its axis are different. And the simplest way to represent that is called resistive force theory, which says it's an approximation of the full slender body theory, which is a theory that derives the flow due to the motion of slender objects in fluid. But that resistive force theory is a local relationship that says that the local force density acting on the fluid is proportional to the local velocity of a filament in the fluid, and the coefficient of proportionality reflects this anisotropy of motion along the tangential, along the axis of the filament versus perpendicular to it. And so I wanted to talk about elastohydrodynamics, and I've told you a fair bit about hydrodynamic forces. And now I need a word of comment about elastic forces before I show you an interesting example where these two work together. And so, to put it shortly, well, elastic deformations cost energy. And so, and if you have a slender filament, there is essentially three modes of deformation, or two modes of deformation that you can induce. If you have an elastic string, you can either bend it or you can twist it. And you can bend it in two directions, and therefore there are two curvatures, so in two perpendicular directions, and there is one quantity called twist that controls how much it's twisted. And you can ascribe energy to these deformations, and in the simplest case, we can say that the energy of bending is proportional to curvature squared. So this is the Euler-Bernoulli theory that if the energy is proportional to curvature squared, then the bending moment will be proportional to the local curvature. And so this will be fully analogous to a spring where the force is proportional to extension and the energy is proportional to extension squared. So here we have two curvatures, so two directions of curvature and one direction of twist. So all of these enter the energy functional for a slender filament in the quadratic form. And then you can have an internal tension along your string, which will contribute to the total energy. Okay, so we've come to the bit where it's time for an elastohydrodynamic example. And I have two. So one example is not mine, but it's the motion of an optimal active cilium. So equipped with this knowledge, you can ask the question of what is the optimal motion, so motion that generates maximal propulsion with minimal energy expense, well, under some constraints, of a single cilium. So I told you that these cilia move in this sort of a two-stroke manner, so there's a power stroke and a recovery stroke, and you can build a simple model based on Kirchhoff equations which are elasticity equations for rods. And the ingredients of this model is that we have active driving forces distributed on these filaments. So this is because of there are dynein motors distributed on these filaments that generate active deformations. which induce internal tension and elastic torque along the filament, which in turn induces hydrodynamic drag forces. And because swimming, or pumping a fluid in this case, is a force-free phenomenon, these forces and torques have to balance. So elastic and hydrodynamic forces and torques have to balance. And then it turns out that for a given filament stiffness, so for a given geometry, there's one parameter that determines the motion, and this dimensionless parameter is called the sperm number, which controls the mutual influence of drag or versus stiffness. So the drag coefficient of the filament versus the stiffness or the bending rigidity of the filament. And the beating pattern depends primarily on the sperm number for the cilium. And it turns out that the model that represents the elastohydrodynamic optimum, or the optimal efficiency, resembles these experimental beating patterns that we see in nature. And another example I want to devote more time to is this paper we published on rechargeable self-assembled droplet microswimmers driven by surface phase transitions. And I'll try to decipher every word in that title now. And so we start off with a very simple mixture of microscale droplets. So these are going to be alkane oil droplets suspended in water. So with a bit of, and then in that water, there will be a bit of a surfactant. I'm a theorist, so I won't care about the details of these systems, but it's a particular oil and a particular surfactant. But what we essentially have here is a microscale droplets. And what we do, we're going to keep the system at the temperature close to the freezing temperature of the surfactant, and we're going to lower the temperature slowly. And so you see the scale bar of 20 microns here, so they are about 40 microns in size. And now we lower the temperature, and they start doing something funny. So they not only start moving, but they produce these, I'm not sure whether you can see them clearly, but they produce these intricate patterns, some sort of tails. Now let's have another look at a different experiment. Well, first the droplet becomes non-spherical, and then it starts producing these tails.
There's not a temperature gradient, right?
No, the temperature is uniform across the system, but we're cooling the system down by a couple of, well, say 5 kelvins throughout this whole video. And so they start swimming, and we asked ourselves the question, what's going on here? And it was known before that this transition from spheres to these hexagonal patterns, this is controlled by the phase transition of the surfactant. So what happens here, I told you that we are close to the freezing temperature of the surfactant, where the surfactant wants to create flat surfaces and straight edges. And so if we are getting closer and closer to the freezing temperature of the surfactant, it takes over from the surface tension of the droplet that keeps it spherical, and the energy contribution from the freezing surfactant takes over. And so the favored shape becomes the first polyhedral shape, but if you keep lowering the temperature, the droplets will flatten out, form these hexagons, and then turn to these rhomboidal shapes. But then if you keep cooling the system down, these droplets become flatter and flatter, but at some point they cannot get flatter, and so they get rid of excess material by producing these tails. And these tails if you take a cross-section of this very thin tail, on the outside it's a plastic rotator face of the surfactant, and on the inside is this alkane oil. So these are tiny tubes filled with oil. And you might get, well, depending on the experimental conditions, you might get 1 tail, you might get 2 tails. I think we have seen 3 tails, but not more. But then they start moving, and we wanted to understand this motion, of whether this can be used as a mechanism for inducing motion. And so we propose a very simple elastohydrodynamics or hydroelastic model for this motion by looking at the motion of a fluid, of a filament that we are pushing into a fluid. We are pushing a filament into a fluid at a constant speed. And so in order to build an elastohydrodynamic model, we need 2 components. One is the elasticity of the filament, and the other one is hydrodynamic forces. And so the cooperation or the coexistence of these forces will hopefully shape the dynamics. And so we start with the first ingredient is ascribing some elastic energy to the filament. We're going to analyze a 2D model here. So our filament can bend, and so the bending energy is proportional to curvature squared, which is incorporated in this term, and can have a tension along its length. And so the total elastic energy is the sum of these two contributions. Now, so from that energy functional, you can derive the elastic force density. And so it has two components. One is the Euler-Bernoulli beam theory component, and the other one is the tension along the filament. Now, the drag force density on this filament is proportional. So this is the approximation of resistive force theory, where the hydrodynamic drag force density on the filament is proportional to its local velocity. And the coefficient of proportionality reflects the fact that such a slender filament is hydrodynamically anisotropic. So it's different drag force along the filament perpendicular to it. And so we have two drag coefficients that are related. And now we need to add a kinematic boundary condition that we are pushing the fluid, the filament into the fluid at a constant speed. So the total velocity of the filament, or the position of the filament in the velocity of the filament in the fluid, has two components. One is the extrusion speed in the tangential direction. So T is the tangent vector of the filament, and the motion relative to the fluid. And we close off this system of equations by imposing the force balance condition, because as I said before, it's Stokes flow, so all the forces acting on the filament have to balance at every instant. And so that allows us to write quite ugly equations for the filament internal tension and for the tangent angle of the filament, which can be translated to curvature. I don't want to spend too much time on these equations, but they're ugly enough to be solved numerically rather than analytically. And so if we do that, well, before we do that, I want to draw your attention to the fact that there was a beautiful experimental paper from 2014 where Gosselin and others, they pushed a piano string into a tank of glycerin. So as to see this motion under low Reynolds number conditions. And you see when you're pushing this piano string into the fluid, initially it's being straight, but then it buckles. It buckles because there is too much tension in the filament, and so buckling is a stress release mechanism there. And so if we code in our equations, this is the dynamics that we see. Our filament initially is straight, we keep pushing it, and so it buckles, and it produces these wonderful sinuous shapes that very much resemble what we see in experiments. And so we think that the patterns that we see here are a result of competition between elastic forces and hydrodynamic forces on filaments that are being pushed at a uniform velocity into the fluid. And so if we look at the question. Yes.
Your simulations were in 2 dimensions.
Yes.
Can you go back, please?
Sure.
The filament is able to cross on itself. How is that allowed?
So in here, this is
in experiments. It's okay. It's not really crossing over.
Yes.
There.
So here we include no self-interaction of these filaments. So
it's a resistive force theory.
Yes. So the different bits of the filament don't really feel each other.
What is the the range of bending rigidities that you have chosen here? Because if you want to do match with the experiments, I'm not sure whether you can model this as a elastic filament.
Well, that was our assumption, and we wanted to see how far we can get with this assumption. And I'll get back to this question because I don't have it off the top of my head, so I'll have to check. But then you see that if you look at the forces and the torques, so we see shapes that are somewhat similar. But we didn't want to compare the shapes, but we wanted to look at the proportion of these droplets. And so if you look at the force and torque these filaments exert at the base, so at the extrusion point, then you see that initially you have an increase of the force, a buildup, which is related to the fact that it's straight and it's getting longer and longer in the fluid. And at this point, it cannot sustain this motion, and so it buckles. And when it buckles, we have, the axial force on the base decreases and starts oscillating around the fx value, and then a torque appears and the shear force appears on this at base. And so this inspired a very simple model for a swimmer where we consider a droplet, and instead of this tail being extruded at this point, we replace it by a force and a torque. Just a pair of force and a torque. And this allows us to find a scaling relationship between the swimming speed and the filament extrusion speed that is given, well, if you rearrange it, the swimming speed is proportional to the extrusion speed to the power 2/3 divided by the size of the swimmer. And this seems to hold very well in experiments. And so we have a very simple model that seems to be good enough to grasp the swimming motion of these spheres. And moreover, you can track these droplets, and particularly droplets that have two tails. And you see some of them, like this one here, they wiggle a lot because these tails seem to be being pushed out of phase. Whereas in this one, they are being pushed in phase, and most of the time, this one is moving along a straight line. And so that inspired us to propose a two-tailed droplet swimmer. Well, this one extrudes its filament in phase, and so it's moving along a straight line. And this one wobbles a lot. And so if we apply our model to a two-tailed swimmer, so replacing each of the tails by a pair of force and the torque, well, this model leads to the semi-empirical model also predicts the correct behavior of these droplet swimmers. On that occasion, we see no hydrodynamic synchronization in this system, which we would see in a biological system. So it is inherently different. But maybe the most fascinating aspect, and I'm getting to the end, of these droplets is that if you cool them down, you see the formation of these tails. So we go through the phase where the droplet gets polygonal, and then these tails are produced. But now we're going to reverse the cooling to heating. And so we're going away from the freezing point of the surfactant, and you see that these droplets, it's a completely fascinating way, but they suck the tails in. And so they're back to square one again. And so they never get tired if we can drive it by temperature oscillations in the range of about 5 Kelvin. And we've done that 40 times, and we keep seeing the same behavior. So they are inherently rechargeable.
Did they go back to the same place?
Not quite, but almost. Elasticity breaks this kinematic reversibility. So they don't really go quite to the same place.
So do you have a control over how many, like, for example, here, all of them form the same shape and one tail?
Yeah.
Like, do you have control?
So the number of tails, this is dependent on the cooling rate in your experiments. So you can, if you're cooling them too fast, you will get more tails typically.
But do they still come back to the similar position if your cooling rate or heating rate is high?
Well, not quite, but almost. So it's not reversible because there's many of them, so they also interact by these tails. If you had one, it goes almost back to the same place.
So the pictures you showed,
Yeah.
you go from a sphere to a triangular like shape. Is the temperature dropping as you go across there? As it's changing shape, is the temperature changing?
Yes.
So what happens if you stopped it in the middle where you had some of those other shapes? Would they be stable?
They're happy with their shape at that moment. So they don't. If we stop cooling the system down, they also stop producing these tails. So if we stop them midway through producing their extrusion process, these tails just stop.
Basically go from a stable to an unstable solution.
Yeah. And so, right. And so, so to conclude that, I wanted to, in this sort of broad introduction, I wanted to convey the message that microscale swimming is in fact about the balance between propulsion forces and viscous fluid drag forces. And it all happens at a low Reynolds number environment. And so because of that, there are some universal rules that all these swimmers, whether biological or mechanical, have to follow. And some of these mechanisms involve elasticity, and then the behavior is determined by the sperm number, so by something that measures the relative importance of elasticity versus drag in the flow. And for an exemplary artificial system, I've shown you these rechargeable droplet microswimmers that are driven by a phase transition of the surfactant that is in this aqueous solution. And so that controls the stiffness of the surface of these droplets. And as a side effect, they produce these tails. This system is fairly simple because it's a 3-component mixture and it's very inexpensive. What is nice about it is that it's completely biocompatible. So you can mix them with natural swimmers. And since most artificial propulsion mechanisms involve some sort of a toxic component, this is a step in the direction where we can mix biological swimmers with artificial swimmers and control and see the collective dynamics that emerges. And another message is that we can get quite a long way with a very, very simple elastohydrodynamic model that quantitatively captures swimming speed in this case. And so this was this droplet project was done in a large collaboration with most of the experimental work done in the group of Nikidenkov in Sofia and Stoyan Smokov in London, and the theoretical team based. We started this back when I was in Cambridge with Eric and Gabriele. And so with that, I'd like to thank you for your attention.
Thanks a lot for being on time. I think I scared you a lot.
No, it's okay.
I gave him a warning that he should.
No, I hate talks that are too long,
Yeah. So we have some time for a few questions. Just go ahead and speak up.
So why does it get extruded in the first place? Is that this as you cool down this crystalline surface surfactant gets smaller and smaller and push the fluid out? Is that what it is? Or
so my understanding of it is that this surfactant creates these molecules of the surfactant align more and more, so the droplet is flatter and flatter, because ideally it would have it's initially a spherical droplet that flattens into something that's semi 2D, and at some point it wants to flatten out, but it can't. So this is again a release mechanism. So one way of dealing with that is to produce these tails. So it's getting rid of excess material from the inside of the droplet.
So you mentioned the reversibility there. So when it is pushing the thread out, or whatever that thing, it is in buckling mode because of compression.
Yeah.
And it has that spiraling motion. So it moves, let us say.
Yes.
Now it starts pulling it in. That is the tension mode. So it will have less buckle. So in fact, there should be huge difference between, it should not go almost back to its original position, if I think about it naively, but you said it almost goes back to the same.
It almost does.
But
so this is not reversible, this motion.
And it's very different. So I would think that it should not be almost at the same position at the end. Was it tested in experiments?
So we never tested systematically how far they go, but in these experiments, they don't go very far, so that we weren't tempted to.
So the original motion was not too much, that's what you're saying.
So another thing that happens there is that these tails get, that's a detail, they're not quite the same elasticity along the filament. So the further you go, the stiffer it is. And so there is some non-trivial melting kinetics of that surfactant when it gets sucked in. But you're right that it's a very different motion when there is buckling mode one way and basically no buckling in the other direction. But that seems to have a minor effect on their propulsion.
So this wasn't the main focus of your talk, but you did discuss the sperm number and you mentioned that there's optimal efficiency you can try to compute if you have both drag and stiffness effects. But I'm curious, I guess, what the assumptions are there. Is the dissipation only due to drag, or is there a model for dissipation within like whatever is generating biological elasticity forces?
I mean, that's probably a long-standing question of what what is the notion of optimality and what is the notion of efficiency in such systems. So in that work of Eric and Christophe Eloi, they measured efficiency by how much power is dissipated by the filament. So there is no notion of biological efficiency. So this is purely mechanical efficiency. But what should really be optimized, I don't think anyone knows in this sort of a system.
I have a quick question, just a clarification. So you mentioned for the filaments that, okay, there's the bending and the torsion. So is it known biologically, does the torsion ever play any role in the deformation of biological, flagella, etc.?
So another example I don't have time to show you is the work we did on the dynamics of DNA. And in the DNA, you have some frozen twist. And in fact, it turns out that the fragments of DNA we typically think, so the double helix structure, is not what the DNA in cells looks like, because in cells, this double helix structure is on top of that is twisted additionally, and it forms these super cold structures that look like a phone cord. So DNA in cells is not only, it has this secondary twist. And so their twist does make the difference because it shapes it determines what shapes the DNA does take, and that in turn determines its biological affinity. So some enzymes do not like to connect to the DNA that's with a particular shape. So twist controls biological function of the DNA to a certain degree.
But swimming, we don't know.
I don't think I've seen an example of a.
Just asking.
No.
If you're you wanted to know what the torsion or the twist, that there are two different things. That this thing is twist.
When the filament,
A twist results in torsion.
Yeah.
Yeah, but in your model, when you're modeling it like that, you cannot really have a twist, right?
No. In the 2D model, we don't have a twist.
Even with 3D, if you model it like a line, then you cannot have a twist.
Indeed. You have to control the Darboux vector.
Then you have to go to Kirchhoff equations.
Yeah, so that's not something we did here. But in general, you can have a general elastohydrodynamic model that includes all three modes of deformation, so both directions of the curvature and the twist. But then it's more complicated because you have to trace the material frame along the filament.
Yeah.
Yes.
But I think there are papers where they see that just a twist can induce swimming.
Yeah. I was just curious that, you put it there, lesson learned, don't put something that
No.
cause more questions. So anyways, any other questions?
So you mentioned that this was biocompatible, at least from the perspective of the oil, the water, and the surfactant. Is it also biocompatible from the temperature perspective? What's the temperature?
Oh, yes, absolutely. Sorry. Thank you. I forgot to say that because this all happens at maybe not room temperature, but maybe room temperature in Hawaii. So, yes, it's in the range of 35 degrees Celsius, which means they are still biocompatible.
Yeah, so just cooling down a couple degrees, basically.
Yes.
Let me understand that better, but this is the way I understood it, it's an equilibrium phenomenon, as in the sense that you cool down and whatever temperature you have, it corresponds to an equilibrium shape of this thing, which corresponds to a certain length of the extruded rod. So if you then keep it at whatever temperature, it's going to sit there. So it's really only moving as long as you change the temperature.
Yes,
Right?
but it's.
So whereas whatever biological thing you have in mind, it would move based on energy expenditures.
So this all happens within a very sort of narrow range of temperatures. It's a couple degrees Kelvin So in terms of what biological organisms would do in the same temperatures, they wouldn't be too affected by this little change of temperature. I'm not sure whether I understood your question very well, but
I don't think it's equilibrium. No?
I mean,
So almost
I would say it's non-equilibrium in the sense that it's driven by the change of temperature.
But it's an adiabatic change. I mean, there's no.
Yes,
It's just that for each you could plot it sort of as a function of temperature,
Yeah.
you could plot the length of the rod. And as you go up and
Yes.
down in temperature, it would always trace the same length.
That we haven't checked, but probably.
And so, I mean, in any case, as soon as you stop,
Yes.
the length of the rod is limited by the range of temperature that you can change.
Yes, so I would say it is non-equilibrium, but it is adiabatic because these is driven by tiny changes in temperature.
How fast do you change it? Are those movies like,
Degrees of a Kelvin per minute.
okay?
These are slightly sped up, 20 times or so.
Great, we had a lively discussion here. Any more questions? No, thank you so much.